What's up, everybody? So in this video, we're going to be talking about enzymes. So this is the HL1, and this one you guys have been waiting for so long. I'm sorry I'm so slow, but it's finally here. So let's just get started. Now for HL, there's a lot of extra little details we got to cover. For example, non-competitive inhibition, competitive inhibition, and a few other little things. So before we get into those nitty-gritty details, let's recap real quick one key thing about enzymes. Remember, enzymes are biological catalysts, meaning they speed up reactions happening in living things, biological things. They're like little tools that help do specific things and help them happen more faster and more efficiently. Okay, now remember that enzymes are almost always made up of proteins. Almost always. What are proteins again? Proteins are made up of a chain of amino acids. These little circles are amino acids. Now that chain of amino acids, that protein, will fold into a specific shape that allows it to do its specific little function, okay? That's important. Just like this little tool here has a specific shape to do its specific function. So that's the quick recap on enzymes, okay? Now the key thing here is that we need to know next is, where are enzymes found? The answer is real straightforward, guys. It can either be found inside or outside a cell. And you're like, duh, of course. Where else can it be found if it's not inside or outside a cell? Yeah, right, it's pretty straightforward. Wherever you can think of it, it can be found. But the two categories are intra, inside a cell, or extra, outside a cell. So let's start here with Po, because I'm using this in honor of the recent Kung Fu Panda movie, Kung Fu Panda 4. Okay, so watch that, guys, if you haven't. Let's say Po, right? He's real hungry like always, and he's gonna take this bean bun or this baozi and put it into his mouth, and it's gonna go down his esophagus into his stomach, right? Now, that baozi or that bean bun ain't gonna be digested by itself by some miracle. It needs an enzyme. Now remember, luckily, our stomach, just like the rest of our body, is made up of cells. Now these cells lining the stomach wall, they're gonna make something. They're gonna make little enzymes, and they're gonna secrete them into the lumen, this big empty space of the stomach. And these enzymes are going to have a specific structure to digest his food, okay? So it's going to attack this bean bun and break it down. Now look, these enzymes, where were they? They were outside our cell. These are extracellular enzymes because they are in the empty space of the stomach. They're not inside the cells. They're outside in a free empty space, okay? Outside of the cells. Now, by the way, sometimes they can be free or attached. So in this case, these are free enzymes. They're freely moving around everywhere. In some scenarios, you guys don't need to know specifics. Some scenarios, enzymes might be attached to a cell membrane or something like that, where it's not moving, but it can still do its function if something comes towards it. In that case, it is attached. Okay. Now, what about intracellular enzymes? So let's say his bean bun was digested into very small pieces, and now it can be absorbed into the bloodstream. Okay, the bloodstream, the blood vessels. So when this nutrient gets absorbed into the blood vessels, it can be carried all around our body to all our cells, because our cells needs those nutrients to survive. Okay, otherwise we will die. Okay, so let's say these nutrients come to this cell here. Look at this very basic cell. It's got a nucleus, it's got a rough ER, smooth ER, mitochondria, very basic cell, and huge enzymes. Obviously, guys, enzymes are really teeny tiny, so they're not actually this big, they're not as big as the mitochondria. It's just exaggerated for the sake of this diagram. But these enzymes, look, they're not outside of the cell like these were in the stomach. These are inside of the cell. So what do we call these? Call them intracellular enzymes. Now, where can intracellular enzymes be located? Of course, in a cell, but where in a cell? In the cytoplasm, inside the nucleus, inside any other organelle, maybe even on in the plasma membrane or the cell membrane. Anywhere in a cell, that's where they can be. And again, they can be free or attached. So these red guys here are free, floating around, but some enzymes, for example, might be inside the plasma membrane, kind of stuck, not able to move. But when the substrate comes towards them, they can still catalyze a reaction, but they were just attached. They're not able to move. Okay, now here real quick, you guys need to know two examples. So you guys know cell respiration, right? This is the process by which we break down our food to make ATP, right? The currency of the cell, the money of the cell. Now, it has very many steps and stages, but you guys will learn all about that in cell respiration. So for right now, you guys need to memorize this. There's no other way, because you guys will learn the real stuff later, okay? So just memorize these two things I'm going to tell you now. So the first step of cell respiration is glycolysis, splitting of glucose. Glyco means glucose, lysis means splitting. It's the first step, and it happens inside the cytoplasm. So the reason why I'm telling you this is because we have to give you an example of where these intracellular enzymes can be. So they can be in the cytoplasm, and one purpose of them could be glycolysis, which is one of the steps of cell respiration. Another example of intracellular enzymes can be inside organelles, as I said. For example, in the Krebs cycle. You know the Krebs cycle, that's a later step. So glycolysis was one of the first steps of cell respiration. The Krebs cycle happens later in cell respiration, and it is in the mitochondria, the matrix, this little cytoplasm of the mitochondria. That's called the matrix. And this one will have enzymes there, working there. Okay, so that's important. I should know some examples of these. So now, that's it. Now we got to move on to the next part. So we have so many reactions catalyzed by enzymes, and sometimes they're not straightforward. And you guys actually need to know, sometimes there are many, many steps to making a final product. Just like you, when you do your homework, it's not just one step. You have to work, you have to go towards your computer, open your computer, put in your password. There are many steps. So the same way in our body, to make a final product, to make a final thing, we need many, many steps. So there are two kinds of pathways to making a final product. Okay, linear metabolic pathway and a cyclic metabolic pathway. And we'll see it very clearly now. Look, a linear metabolic pathway, it looks something like this. So you will have an enzyme, and the initial substrate, so the first substrate, will bind to the active site of this enzyme, and this enzyme will catalyze the reaction and make a product, in this case, an intermediate. So this is the product. Now this product is not the final product. It's like an intermediate product. So it's going to now bind to another enzyme, and that enzyme will turn it into a C intermediate. Now that substrate will now bind to another enzyme, and that will eventually make the final product. So you can see there are many steps. It's not just one reaction. It's a chain of reactions. So I need to emphasize here that each enzyme here is different. Because remember, I told you enzymes are specific for each reactant. So this enzyme will be a specific shape for this substrate. This enzyme will be a specific shape for this substrate. This enzyme cannot do the job of this substrate, and of this enzyme. And this enzyme cannot do the job of that enzyme. It's like this tool here that I mentioned before. This tool has one job and one job only. It cannot do the job of a hammer. Okay? So it's the same thing here. That's why I put numbers here. So this is enzyme 1, enzyme 2, because I was too lazy to make them all different shapes, so I put numbers on them. Bear in mind, the numbers means they are different. Okay, cool. But you can see the key thing here is a linear metabolic pathway has a substrate, and then eventually it has a final product. So it's linear, okay? Now, what are some examples of that? Glycolysis. Remember, we just talked about glycolysis, remember? It happens in the cytoplasm. It's one of the first steps of cell respiration, right? That is, the process of glycolysis is a linear metabolic pathway. So it's like this. It's like a few steps. Glycolysis will be a few steps like this to make a final product. Okay? Now, what about cyclical? You can imagine, just like the word, it's like a cycle, but it's very different from linear. Remember, linear started off with an initial substrate and eventually made some intermediates, and then finally there was a final product. With cyclic metabolic pathway, the initial substrate is also the final product. It's a cycle. And you might be like, what's the purpose of this, my guy? You're right. Right now, you're not going to get it. But when you guys learn about specific examples of cyclic metabolic pathway, like when you learn about the Krebs cycle in cell respiration, or when you learn about the Kelvin cycle, those are all cyclic metabolic pathways. And it may look from the surface that it's useless, but actually, when the reaction happens, so let's say A gets turned into B, there are other little mini byproducts or mini products that are made that are useful to the cell. So maybe turning A into B isn't useful, but to turn A into B, a little other byproduct was made, and that product might be useful. Okay, so don't think of cyclic metabolic pathways as useless and linear metabolic pathways as useful, because linear makes a product and this one doesn't really make a product, okay? Because it doesn't work like that. When you guys really learn an example, it'll be far more clear. Just know it is useful, but the initial substrate is regenerated, okay? And you'll see why it's useful when you learn about specific examples in later chapters. But two examples you guys should know is the Krebs cycle, also known as the CTA, or the citric acid cycle. It's part of the cell respiration. And Kelvin cycle, which is a cycle that happens in photosynthesis. Okay, awesome. So quickly, key concepts. Different pathways have different lengths. They're not always 4 things long or 5 things. It could be 10, could be 2, different lengths, okay? Each reaction requires its own unique enzyme. So this was a unique enzyme to turn A into B. This was a unique enzyme to turn B into C. They're all different, okay? Each enzyme is specific. I cannot overemphasize that. So cyclical pathway begins and ends with the same substance. And then lastly, the final product of one pathway could be a substrate of another. So don't think of everything as separate. Our body is so complex and interwoven. Look, it can even be like this. Okay, so say we have a linear pathway here, starting with A and ending with C. This final product could have a job, but there could also be scenarios where this product is simply going to go and become a part of another pathway. For example, look, let's say that final product starts joining this one. That final product was the initial substrate of this pathway. Okay, so you don't think that all final products are final products. Some final products are going to join other pathways. Okay, just like this. So this final product of the linear pathway became the initial substance of this cyclic pathway. Okay, that's important. The body is pretty complicated, unfortunately. Now, when we learned about the core content, the SL/HL video, we learned a lot of information about enzymes and how they normally work, how pH can affect enzymes, how temperature can affect enzymes, and so on. Now we need to learn about a little molecule called an inhibitor and how inhibitors can affect enzyme activity, right? An inhibitor is a little molecule that can bind to an enzyme and decrease its activity, making it not work as well as it normally should. Now, there are different ways in which inhibitors can act. The first way we're going to learn about is called competitive inhibition. And then we're going to learn about some other ways that are similar, but they are different, okay? There's some subtle differences. So let's learn about competitive inhibition. So let's explain in this little box. So here we got what? We got our enzyme, okay? And then we got what? A substrate. And what does the substrate like to do? It likes to bind to the enzyme's what? Active site. And then the enzyme will catalyze it to make a product. Now, in competitive inhibition, there's going to be a specific kind of inhibitor that's going to competitively inhibit this enzyme. What on earth does that mean, my guy? Here's the inhibitor. Okay, we call this inhibitor a competitive inhibitor. Why? Because it's going to compete with the substrate for the active site. Remember, the substrate wants to bind to this active site. Now, this competitive inhibitor is going to be a pain. It's going to go and try and compete with that substrate for the active site. It's going to try and get there first. And the thing is, when this competitive inhibitor binds to that active site, now the substrate can't bind there anymore, which means the enzyme cannot catalyze that substrate and make a product. So right now, this enzyme is just being blocked up and no product can be made. So that's why it's called a competitive inhibitor, because it's directly competing. They're like pushing each other. No, me, I'm going to get it. Me first. So they're directly competing. They're competing with each other directly for this seat here. Kind of like musical chairs, right? Where you try and get to the seat first. That's how a competitive inhibitor works. Okay. So now this guy cannot bind there no more. No product made. Unfortunate. Now, one very important thing guys need to know, this is super common in multiple choice questions. Competitive inhibition is reversible. What does that mean? You know when you go to the cafeteria and all the seats are taken, you're like, oh, I can't eat. All you have to do is wait a little bit. And eventually those people inhibiting, those competitive inhibitors taking up your seat, they will get up and leave. When they leave, guess what? You can go and take your seat again, right? So it's the same thing. These competitive inhibitors, when they bind to this active site, blocking the substrate, they may leave. It is reversible. They can detach and the substrate can bind again. So just think of that cafeteria example and you're fine for this. Okay, it is reversible. It's not permanent. Awesome. Let's look at some keywords, guys. So the competitive inhibitor competes directly with the usual substrate for the active site. It has a similar structure, not the exact same necessarily, but similar enough that it can bind to the same active site as the substrate. Okay, awesome. Now, here's the next thing. You guys need to know how to recognize a certain curve when it comes to competitive inhibition and also the other kinds of inhibition. Now, it might look like hell at first, but guys, it's going to make sense. And if you understand it, you'll never need to memorize anything about this again. So let's look at the curve. Very simple, start off nice and straightforward. We got the x-axis, substrate concentration, just meaning how much substrate we have, starting from very little substrate to a lot of substrate. Whenever you see these square brackets, it just means concentration, guys. So if we have an S here and the square inside the square brackets, it means the concentration of S, which is substrate. Now, then we have here the y-axis, which is reaction rate. So how much product is able to be made, how much product this enzyme can make. So you can see two lines on here. First, we can see the normal one. This is the curve that is made when there is no competitive inhibitor or no inhibitor at all. There's just an enzyme and a substrate, okay? That's the curve made. And then we can see here the curve for competitive inhibition. You can see they're slightly different. So to understand the curve for competitive inhibition, we must understand the curve for no inhibition, because most people are already confused when they see this curve and they think, okay, why is it go up like this and then reach a plateau? Why isn't it like linear? Why isn't it just straight up all the way? Let me show you. We need to understand this, then everything will be so much easier to remember. So let's look here at some place in our body. Let's say we have some enzymes. Okay, look at these enzymes. Let's say we have no substrate at all. Okay, so if we have no substrate, can these enzymes make a product? No, right? So the reaction rate will be 0. So that's where we start here. Now let's say we increase, okay, the substrate. Let's say we go to here. We now have this much substrate, okay, this much. Let's say we put some substrate. Now what's going to happen? This substrate can go around, it can eventually collide with the enzyme, and then this enzyme can catalyze the reaction to make a product, okay? So with increased substrate concentration, what do we have? An increased reaction rate, because we were able to make more product. The enzyme was able to make more product with more substrate. Okay, so what do we have? We can say we have more increased reaction rate compared to the beginning when there was no substrate. Now let's increase the substrate even more. Okay, let's say we have more. Now, how's the reaction rate now? Because now we have much more substrate. We're more like here now. We increase the substrate even more. So now you can see there's a lot more substrate. When they move around, the reactions many more reactions will happen, because this one can bind here. Maybe this one will eventually collide here and make a reaction. Maybe this one will collide here and make a reaction. So by increasing our substrate concentration even more, there was even faster reaction rate, because we were able to make even more products in the same amount of time. Okay, because the enzymes were all busy now, instead of before where only the one was busy. Now they're all busy. So the reaction rate is, of course, much more. Cool. Okay, so the reaction rate's even more. Now, what if we increase it even more? Let's go crazy. Increase it a lot. What's going to happen? Let's increase these guys. Okay, so we increased them a lot now. We got a lot of these substrate. What's going to happen now? What's going to happen to the reaction rate? Do you think it will increase? Actually not. Because think about it. These three enzymes are already busy with a substrate. So now if you put even more substrate, they can't do more than that, because they're already saturated, they're already occupied, they're already busy. So even if you give them more work, they can't do that work because they're already full. So even when we increase the substrate concentration even more, the reaction rate will not increase much. It'll increase a little bit. It will start reaching a plateau. It will start kind of going flat, because the enzymes are all already occupied. If you give them even more substrate concentration, they can't work faster. They're already so busy. They're already as busy as they can possibly be. So this explains why this curve increases initially fast and then it starts slowing down, because near this stage here at very high substrate concentrations, these enzymes are already very full. They can't do any more work, okay? They're saturated. Okay, awesome. So that explains the normal curve. The normal curve with no inhibition. Okay, so now let's have our enzymes here. Okay, our substrates. Now we're going to explain why the competitive inhibition curve looks like this. So we have our three substrate, we have our three enzymes. Now we bring in Mr. Inhibitor, the competitive inhibitor. Look, so now remember, when we had three substrates, the rate was like around here. It was pretty fast. Okay, it was pretty fast. But now we have three substrate, but we also have inhibitors. So how's it going to happen now? Let's say now the reaction starts. These guys will try and go for the active site, but maybe an inhibitor will get there first and block it. Now these substrates can't bind there, which means now this reaction can't happen, so the reaction rate is a bit lower. So in the presence of competitive inhibitors, the reaction rate will always be lower. Maybe some substrates can find a spot and make a reaction happen, but maybe some are blocked, so they can't get the reaction to happen. So you can see in the presence of competitive inhibitors, at the same concentration of substrate, so three of these substrate, now the reaction rate is lower. See, before without inhibitors, it was very high. Now at the same substrate concentration, it's a bit slower. Okay, awesome. That makes perfect sense, right? But now the tricky thing is, why on earth, if we increase the substrate concentration, does the rate of the competitive inhibition eventually become the same as with no inhibition? Doesn't that not make any sense? Like, why? Because this one has inhibitors. So how on earth can the rate increase up till this normal amount with no inhibition? How is that possible? That's a great question, but look, it makes perfect sense. So let's say we have our two inhibitors here. Okay, we got our enzymes. Now, let's increase the hell out of this substrate concentration and see what happens to this curve. Okay, so we have competitive inhibitors here, but let's increase the substrate concentration a lot. Okay, we got a lot. It's going to be a bit messy, guys, but we're going to do it. Okay, look that. We have a lot of substrate now. Let's say the reaction happens. Look, these guys, they're trying to get to the active site, okay, of these enzymes, but there's so much substrate that the substrate almost always gets there first. The substrate is almost always in the way. So because there's so much substrate, it's so rare for these competitive inhibitors to actually get to a active site to block a reaction. So at very high substrate concentrations, the reaction rate is exactly the same as if there was no inhibitors. Even if an inhibitor gets to bind there, remember, it's like the cafeteria scenario. Eventually it will get out because it's reversible. And when it gets out, that substrate will just bind and the reaction will be like normal again. So at very, very, very high substrate concentrations, this curve will eventually reach the same level, the same maximum reaction rate as the no inhibition curve did. Because these, as you increase the substrate concentration, they overwhelm these competitive inhibitors and almost make it seem as though they're not there. They're physically pushing them out of the way the whole time. It's almost like they're not there. Okay, so I hope that makes sense. So here's a few things you guys need to know. You can read this by yourself. All of this stuff is quite straightforward. It's kind of what I said. So the rate is reduced, okay, but at very high concentrations, the maximum rate is still achieved, okay, because they're outcompeting, the substrate is outcompeting all of these competitive inhibitors, okay? Of course, if you add more enzymes, then the rate of reaction can increase more, because the more enzymes you have, the more substrate has space to bind to, right? But that's straightforward. That's not too important. Okay, now you guys need to know a real example before we move on to the next one. So let's get rid of this mess real quick. Okay, so you know in our bodies, guys, this is actually very important, a real example, because we have now the theory. Let's get a real example down. You know, in our body, we have a liver. It's an organ, and in this organ, there's a lot of cells that has enzymes that like to bind to a substrate and make cholesterol. Okay, so for example, this enzyme will bind to a substrate, and cholesterol will be the product. Now, cholesterol is good for you. It has a lot of functions. Right? We learned about cholesterol. It can be in your cell membrane and help with fluidity. It has a lot of important functions. But at very high concentrations, cholesterol is very dangerous for your heart. You can get heart attacks and other heart problems. Okay, you guys don't need to know the details about that, but high cholesterol can cause heart problems, okay? Like heart attacks. Now, so doctors, for patients with very high cholesterol that are at risk for certain heart attacks and problems like that, they will give them a certain medication that is a competitive inhibitor, okay? And this specific medication is called a statin. Now, this medication behaves like a competitive inhibitor and will basically go and bind to these enzymes that catalyze the production of cholesterol. Because when these statins competitively inhibit these enzymes, then these substrates cannot bind there anymore temporarily, and so there's a reduction in the rate of cholesterol production. So thereby, taking these medications, those patients can reduce their cholesterol level and kind of improve their health a little bit. So that's very, very important. So that's a real example of a competitive inhibitor used in medication. Now let's move on to the next one. We just covered competitive inhibition. Now let's do non-competitive inhibition. Another name for this one is allosteric inhibition, and we'll see why this name makes any sense. So the difference with this one is this. Here we have our enzyme, okay, and we have our substrate. Now, the inhibitor in this case behaves differently. In competitive inhibition, the inhibitor did what? It competed for the active site, okay, of the enzyme, and therefore thereby blocking it from the substrate. Now, in this case, our inhibitor is not going to bind the active site. It's going to bind another site on the enzyme called the allosteric site. So this is not the site where the substrate wants to bind. It's another site on the enzyme. Now, when this inhibitor, this allosteric inhibitor binds, it's going to change the structure of the active site. That's crazy, right? Just like this. Look, when it binds here, look, the structure of the active site changed. Now, what does this mean? this means that the substrate is not going to fit well here anymore, okay? Which means a reaction cannot happen. The enzyme cannot make a product, okay? So this is why it's called allosteric, because allosteric means allo means other, and steric means shape. So this inhibitor causes a change in shape of the active site. That's what's called an allosteric inhibitor. It changed and it makes another shape. That's pretty cool. So let me show you here. And very, very important, just like competitive inhibition, remember this one was reversible, meaning eventually this competitive inhibitor can detach and the substrate can bind. This one is also reversible. Look, when this allosteric inhibitor detaches, the shape will come back to normal and the substrate can bind again and the product can be made like normal. So it's also reversible. Remember that, for the love of goodness. Okay. So here, the non-competitive inhibitor instead binds to the allosteric site. It causes a change in the shape, conformational change of an enzyme's action, making it non-functional. Okay, so just like the other one, we need to look at this curve. Here we go. We all know why this one looks normal, the no inhibition curve. Now, why does this one? Look at the difference. Compared to the competitive one, it looks different. The competitive one had a reduced rate, but eventually at very high concentrations, remember, the rate reached the same maximum as the no inhibition. But this one, it has a reduced rate, but even if we increase the substrate concentration, the rate never reaches the same maximum as the no inhibition. Why is that? Very important question. Okay, here we go. So here we have our enzymes, and they can bind, and product will happen. Okay, but look. When we have an inhibitor, look what happens. Let me rotate it for you guys, for the cringe people. Okay, so remember, when this guy binds, what happens? The active site changes shape, and now the substrate can't bind anymore. Okay, so let's say, look, let's put another one. Let's say there's another inhibitor. Now, look, now these guys can't react. Now no product can be made. So let's say, even at very high concentrations, look what happens. Look, we're increasing the concentration. Does anything change? No, because they still can't bind, because the shape is changed because of the allosteric inhibitor. Even if we put more, they still can't bind. So even if we're increasing substrate concentration, the reaction rate can't really increase. Okay, it's always going to be a bit lower than no inhibition. And the reason why is like this, because these guys here aren't trying to compete for the active site. Okay, they're binding to a completely different space. So even if we do this, look, even if we have the two enzymes and we say reaction start, these guys aren't going to try and bind to the active site. So these guys aren't going to block them, because even if these guys come here and they bind, this guy can just go around and bind here. And when it binds here, the shape will change and this guy will not fit anymore and he will detach. So the reason why the reaction rate never reaches the same height is because these guys aren't competing with each other. It's non-competitive. So even at very high substrate concentrations, these inhibitors can just go around and bind to the bottom and cause these guys to not be able to react. Therefore, the reaction rate will never be as high as no inhibition. That is different from competitive inhibition, where these guys are physically competing for the same spot. So at very high concentrations, these guys can just outcompete them, okay, hustle them out or out of the way. Okay, so that's very important. So you need to know the differences between those curves. So same thing, it decreases the rate. The key difference is this one, in non-competitive inhibition, there will be a lower maximum rate, okay? Not like this one where the maximum rate was the same at high concentrations, okay? So even by increasing the substrate concentration a lot, it doesn't influence the rate, okay? The rate will never reach the maximum. It will be lower because these guys are binding to a different spot, okay? Okay, awesome. Real quick, some example you guys need to know is not too important. Metallic ions such as mercury can behave like a competitive inhibitor. So they can bind to this spot here. Remember, it's a protein, so it's made up of amino acids. So these metallic ions like mercury can bind to this area here, to these amino acids here, specifically the sulfides. You know, some amino acids have sulfur groups. So when it binds to those amino acids, it will cause a structural change of the active site. So that's just a small example you guys need to know. It's not too important. Okay. So again, here I have one curve showing all of them. We can see the no inhibition curve. Very, very important. If you get a curve like this and ask you which curve is which, remember, competitive inhibition reaches the top. Okay, it can reach the maximum rate at very high substrate concentrations. Allosteric inhibition will not reach even at very high substrate concentrations. Why? Because the inhibitors don't compete for this site. They bind to a different spot. So even with a lot of substrate, the substrate is not going to be in the way of these inhibitors because they bind to a different spot. They're not competing for the same spot. Okay, cool. So that's very important. This is super important to know about. Okay guys, one more example of an inhibitor. There's not that much you guys have to know for this one. You don't have to recognize a curve for this one. You guys just need to know the concept. So let's take a look at it. So this one is pretty cool. Look at it. We got our enzyme here, and now we've got an inhibitor here. But the difference is, remember for competitive, the inhibitor would bind here, and it could detach and let the substrate bind again later. And then for non-competitive inhibitor, the inhibitor bound to the allosteric site, causing a change in the structure of the active site so that the substrate can no longer bind. Now, for mechanism-based inhibition, the inhibitor will bind to the active site. So it's very similar to competitive inhibition, but the difference is this inhibitor will bind very strong. It's going to make covalent bonding. So this is a permanent bond. So this one is irreversible. When it binds, it cannot detach. So another name for this kind of inhibition is suicide inhibition, because when this inhibitor binds, this enzyme is basically non-functional forever. It basically suicided. No longer going to work. Okay? So the mechanism is very similar to competitive inhibition, but the difference is when this inhibitor binds, it's permanent, unlike competitive inhibition, which is reversible. Okay, so it's because there's a covalent, very strong permanent bond. Okay, pretty cool. So now, look at this. You guys need to know an example. So you guys don't need to know the curve, but you guys need to know an example, real life example. So look here. When we look at bacteria, right? Bacteria, what's different from bacteria? They can have a cell wall. Remember, me and you, our cells, we have a plasma membrane, but no cell wall. So to make a cell wall, these bacteria have certain enzymes. Look, this enzyme, it's got a name, transpeptidase. Remember, it's an enzyme because it ends in ase. Anytime you see ase, you know it's an enzyme. So this enzyme, what it does is it binds a substrate and once it binds that substrate and catalyzes a reaction, that product is going to be a constituent of the cell wall. So it's going to keep doing that, and eventually the cell wall will be nice and strong. Okay, nice and strong. That's very important. Now, very cool. When we get infected with bacteria and we get sick, right? Sometimes our body cannot manage it by itself. So we need to go to the doctor and they'll give us medication. You know, when you get a bacterial infection, your doctor might give you a certain drug. You guys might have heard of penicillin, right? It's an antibacterial drug, an antibiotic. How does it work? You guys are going to learn right here. It's pretty cool. So look here. You know this enzyme, okay? It can be inhibited by this drug, penicillin. Look, here comes penicillin. Your doctor is going to give you this drug. And this drug is going to go do what? It's going to act by mechanism-based inhibition, meaning it's going to go and bind to this active site permanently, okay, of this enzyme. Guess what? When it binds to that active site permanently, this enzyme has basically suicided. It cannot make any more product. Now, guess what? If it cannot make any more product, its cell wall will become weak and start breaking. When its cell wall becomes weak and it starts breaking, it's like your house's walls falling apart. The bacteria just dies. Without strong cell wall, it's dead. Okay, it's going to break. That's going to cause it to die. Okay, so that's very cool. But what's interesting as well is this. Let me show you. Oh yeah, by the way, you guys need to know this. This sometimes comes up. That's the only reason why I'm putting it. The guy who discovered penicillin is called Alexander Fleming. They love to sometimes use this in multiple choice because this guy is super important in science. I know you guys can't care less about this guy, but it may come up in the multiple choice. Remember this guy. This is a pretty freaking big discovery, so that's why they want you to know about him, okay? Now, some bacteria have certain genes or genes that make them resistant, because these genes that they have makes a certain thing. Let me show you, okay? It's a mutation, a specific gene mutation that allows them to make a certain molecule. Let me show you. Now, these bacteria, they can make a certain molecule called penicillinase, okay? So it makes a little enzyme like this, these bacteria. So when the doctor gives you the penicillin, they have this enzyme which they're making that's going to break down the penicillin. That's crazy, right? So therefore, when the doctor gives you penicillin, the bacteria just makes a little molecule, a little enzyme that breaks it down so that the penicillin is useless, and so that the penicillin can no longer bind to the transpeptidase, this enzyme, to stop it from working. So that's a pretty brutal thing, because then even though you're taking penicillin, you won't get better, because this bacteria is mutated. It is unique in the sense that it makes a certain enzyme that can break down the penicillin. See, it's a penicillinase, so it's an enzyme. It makes this enzyme that can break down the medication the doctor is giving you. In that case, you won't get better, so you'll need to go back to the doctor, and they may give you another enzyme, another medication that will target something else about the bacteria instead of the cell wall, so that you can still get better, okay? But you won't know that until after you try to take penicillin and you don't get better. So that's a pretty cool example of mechanism-based inhibition. I hope that makes sense. Okay. I think that's it for that. Now, we're moving now onto something called feedback inhibition. I want you to know this is not a new kind of inhibition. You'll see what I mean by that. We already learned all the kinds, okay? We learned competitive, not competitive inhibition, non-competitive, and mechanism-based. That's the three kinds of unique ways of inhibition, okay? The three different mechanisms. So feedback inhibition is not a kind of inhibition. You'll see what I mean. So we remember how we had a linear metabolic pathway, right? So feedback inhibition is something pretty cool, and I'll show you what it is. So sometimes you have this linear metabolic pathway, which will eventually make a final product, and that final product will have a specific function, right, in our body, whatever it may be. Now, let's say at this moment you don't really need this function anymore, okay? Like, you don't need it anymore. Like, at that moment, you don't need that to happen. It's not important at this moment. So you don't need this product anymore, okay? So what's going to happen is, this pathway is going on and on and on, so you're making a lot of this final product, but it's not being used because you don't need that function at the moment. So it's not being used. This final product is just building up and up and up because it's not being used. So to prevent your body from making too much of this final product when you don't need it, what happens is something pretty interesting, and it's called feedback inhibition, or also called end product inhibition. And it works like this. This product that is being made, that is building up because you're not using it, what it's going to do is it's going to behave like an inhibitor, like an allosteric inhibitor. Remember our non-competitive inhibition? It was called allosteric inhibitor. Well, this is a kind of area where this is used. So this final product will now kind of build up and build up and up, but we don't want to make more of it. So what happens is it will go inhibit one of these very early enzymes in this pathway. It will go there and do allosteric inhibition. Okay, this is the most common way in which they act. Allosteric, so it's going to bind to the allosteric site and stop this enzyme from working, thereby stopping the creation of B intermediate, and thereby stopping C intermediate, and thereby stopping D intermediate, so that this final product doesn't become more and more and more, even though we don't need it. So why is this cool? This is very cool because this prevents the cell from wasting chemical resources, because some of these reactions require ATP to happen, requires energy to happen. So we don't want to waste unnecessary energy to make all of this final product if we don't actually need it anymore. Maybe we need it later, but not right now. So it prevents the cell from wasting chemical resources and energy by making less of a substance that it doesn't need, okay? So it's making less of it because it doesn't need it right now, okay? Now, what if the function returns? What if we start needing it again? So right now we start using it. So we had a lot of them built up, and now the function starts being required again. So they start being used. And now, guess what? Because they start being used, now their number decreases and decreases. And if their number decreases and decreases, now there's going to be none of them left to inhibit this enzyme. And if this enzyme is no longer inhibited, guess what? Now A can be converted to B again because the enzyme works again. So now we can make the final product again and increase the final product again so they can carry out the function. So it's pretty cool, right? So that's why they call it end product inhibition, because the end product that will go and inhibit one of the earlier enzymes. Okay, so let me summarize that here for you guys. When present in high concentrations, and the end product will bind with the allosteric site of the first enzyme, thus bringing about inhibitions. And it will be present in high concentrations when it's no longer being used for one or other reason. Maybe it's not needed in that moment. Then it will build up and build it up and up, and then some of it will go back and inhibit this enzyme so that less of it is being made, so that we don't waste all our resources. When it needs to be used again and it starts being depleted, then there will be no final product left to inhibit this enzyme. So the enzyme will be activated again, so reactivated again, and the final product can be increased again to carry out the function. Now, the next question we need to ask is, why are we inhibiting the first enzyme? Why doesn't this final product go and inhibit this one or this one? It's a very good question, and it's very important to consider. So this is done so that the buildup of the intermediate so that a buildup of the intermediate substrate in the cycle does not occur. Okay, so let's say this guy goes and inhibits this guy, 3 instead of 1. That means A will still be converted to B and still be converted to C. So we will still unnecessarily make B and C. Okay, we're still wasting energy to make B and C. Whereas if he just goes back to inhibit the first enzyme, then we prevent making B and C in the first place for no reason. So it's done so that the buildup of intermediates does not occur. So this can conserve energy since the following reactions will not happen for no reason. So it's very important to know that feedback inhibition typically involves the final product inhibiting one of the very early enzymes, typically the first step in the pathway to save energy. That's important to understand. Okay, let's look at a real life example real quick, guys. So here we got it. Okay, we got, in our body, right? I told you we've got amino acids, okay? We got, from the core video, we've got essential and non-essential amino acids. Remember, the essential amino acids were the kinds of amino acids that we don't make. We need to eat them in our diet. And then the non-essential amino acids were the ones that we can, that our body can make by itself, as in we don't need to eat it, okay? Now, look at this example. This applies to bacteria and plants, by the way, not to humans, because this is a kind of amino acid. Okay, you see this one, isoleucine. It's a kind of amino acid that humans need to eat. So humans need to eat it. Humans cannot make it, but bacteria and plants can actually make it. They can synthesize it. So that's why this is specific for bacteria. That's an example for bacteria and plants, not for humans, because humans need to eat isoleucine. We cannot make it, okay, like this. But plants can, and bacteria can. So how it works is you got another amino acid, 309, and this amino acid, through a series of interactions, will be converted through this linear metabolic pathway to isoleucine, okay, another kind of amino acid. So you can see plants and bacteria are able to synthesize this amino acid from another amino acid. That's pretty cool. But again, same concept as before. When this amino acid builds up, if it's not being used for one or other reason, so it builds up, what's it going to do? It's going to go back to the first enzyme and inhibit it. In this case, what the first enzyme is 309. So 309, the amino acid, the aminase. Okay, it's going to go back and inhibit it by allosteric binding to prevent the unnecessary production of this when it's not needed. Okay, so they also call this negative feedback because this guy is basically, when you do something in your house and your mother or your brother's like, that was bad, don't do that again. This is kind of the same thing. So when the body's making too much of something, it may result in negative feedback where this molecule physically goes back and says, don't make any more, we have enough of you. So it's negative, it's giving negative feedback, saying don't make more, we have enough, negative feedback. There's another kind of feedback called positive feedback, where a molecule will go back and say, make more of me, there's not enough. That's positive feedback, okay? But for now, you should know that feedback inhibition is an example of negative feedback, where a molecule will kind of signal back and say, that's enough, don't make more. That's an example of negative feedback. That's just a small key term you guys should be aware of, okay? Awesome, that's it for feedback inhibition. So I wanted to show you that this is not a unique kind of inhibition. This is just a specific example where allosteric inhibition or non-inhibition can happen in a real example, okay? Awesome. Okay, lastly here, this is very small, not that important. So you know how we just talked a lot about examples of things that can inhibit enzymes, all these non-competitive, competitive inhibitors and so on. There's actually something called a cofactor that is sometimes needed to activate an enzyme, right? We talked about things that inhibit enzymes, but look here. Sometimes an enzyme can bind a substrate, but it won't work until a little molecule or a cofactor, we call it a cofactor. It's a little thing that can bind to this enzyme, and basically it's like an on switch. So without this cofactor, it's not always, only some enzymes need this. So some enzymes without this cofactor will not make any products. But as soon as a cofactor binds, that will activate it, turn on this enzyme, and allow it to actually do its function and make products, okay? So that's pretty important to know about. So cofactors are pretty good. They're basically like the opposite of inhibitors. They can kind of activate an enzyme. So many enzymes need an inorganic ion or a non-protein organic molecule to work properly. These are called cofactors. They can be, examples are vitamins or things like that, okay? So what do I mean by inorganic? So remember, inorganic, we have organic matter or inorganic matter in life. Organic matter includes anything with carbon-hydrogen bonds, okay? These basically are the 4 macromolecules: carbohydrates, nucleic acids, lipids, proteins. Anything inorganic does not have carbon-hydrogen bonds. So these can be minerals like magnesium, iron, calcium, things like that. Those are all inorganic things. So these cofactors, they're typically non-protein things. Remember, enzymes are proteins. They're typically non-protein things, so anything else except proteins, or they can be inorganic ions like magnesium or potassium or things like that. So question time. What is correct for competitive inhibition? Try it for yourself, guys. So the answer here will be the inhibitor resembles the substrate, remember? And it competes by binding to the active site. So A is definitely correct. Let's see why the other ones are wrong. The inhibitor resembles the substrate. That's true so far, but it does not bind the allosteric site. That happens in non-competitive inhibition. The inhibitor doesn't resemble. That's not true. The inhibitor doesn't resemble. That's not true. They do resemble. So the answer is definitely A. In non-competitive inhibition, where does the inhibitor bind to the enzyme? Again, the allosteric site, remember? In competitive inhibition, they bind to the active site. What kind, did I reveal the answer? Oh, no, I didn't. Answer is C. What kind of inhibition is reversible? That's why I emphasized the hell out of that. Remember, both non-competitive and competitive inhibition was reversible. Okay, so the answer is both A and B. Which one is not reversible? We'll go back to that now. Remember, mechanism-based inhibition. This one was non-reversible. Okay, that's important. So that's it for this video. I really hope it was useful. If you guys want awesome, sexy notes and lots of IB-style questions, go check out our website, teachme.org. The link is in the description, and I'll see you guys in the next one.